Hello. So we are now where we could start chapter 7, but I think what I want to do instead is go all the way to the end to this back page, and I want to do the kind of hodgepodgey things we have left that make sense while we're still talking about cells and organelles. So this particular half sheet, I'm going to cut it, and I want to have 2 pages, just so I can write over here in addition to this. So this little table is a summary of the main organelles that you should understand and know what their functions are. I'm sorry, I'm talking over my cutting. And the thing about it is, truthfully, the whole content with AP doesn't really include a whole lot of time for this. They seriously believe you remember something from when you were a freshman. I know, crazy. But, so that's why we have this table. The problem I have is this. Go on the tape. I don't know why. I'm the same way. I'm not trying to act like anybody's wrong or awful, but we don't go back and study things that look like this. We seriously study what we write. So I'm going to tape it in there. It's good for reference, but we're going to, I don't know if this is zoomed in. Is it? I can't tell. Whoops, that's not what I wanted to do. Oh, I am tired. No? Okay, well, let's go with that now. What was I saying? I was saying it's difficult to work with stuff like this. But they do think you remember stuff from when you were in freshman biology. And even if you did, it's not to the detail that we need. So we're going to go through here, and we're going to take just a few minutes, and I'm going to let you know what parts are important, and we're going to move it out of this table and move it over here. Now, you feel free to highlight, but I'm telling you, you probably won't go back and study it that way. So we're going to start with the nucleus. I'll put it over here. And it's giving you 2 parts that are super important. So you need to always speak to structure leads to function. So the structure of the nucleus is a double membrane. It says it's surrounded by a nuclear envelope, double membrane. It's perforated, which means it's punctured, and it has little nuclear pores. The envelope is continuous. Sometimes they use the word contiguous, which is the same kind of thing, with the endoplasmic reticulum. Well, we know that because we drew that, right? Right here it is. There's my nucleus, and I made it continuous with, this is the whole endomembrane piece, the rough ER and all that with the smooth ER and the Golgi. So I'm going to move over here what's the important part about that. So when it comes to structure, I want to remember these things. That the nucleus has a double membrane. We're going to talk about that in just a second when we get to mitochondria and chloroplasts. But it's a double membrane. It's going to have the whole membrane structure of the cell associated with it. So I'm going to write that. An entire membrane structure of the cell is attached to it. You know, sometimes we get kind of crazy with these fancy words. We want to say continuous and all of that. We just need to speak clearly. The membrane of the whole cell is attached to the nucleus. There's a reason for that. It's transport. It makes it all flow better. The actual nucleus itself, it has pores, and they are specifically, they're specifically small. And by that I mean DNA can't go out of them. So they're going to be selective about what can come in or out. Like they're purposely the size they are. Function, it has chromosomes, which are made up of chromatin. DNA and protein. Yes, later we're going to have to get into all of that. Right now it's good enough to just say DNA is in there. It also has a special spot. And you know, when you were younger and you drew your cell, I think we talked about this, and you put your nucleus and you shaded this little thing and you're like, I've got an awesome cell because I put my little shaded spot in my nucleus. Well, nobody knows what that does. But that's the nucleolus, and it has a function. So it's really got a structure, and I'm going to put it right here. Nucleolus is here in the nucleus. So the nucleus is going to, so this is structure, this is function. It's going to house the DNA, and it keeps it there. I've kept saying this is like the reference part of a library where you just don't let something precious get checked out. You don't have the only edition that you have of some famous book and go, yeah, that crazy kid who loses everything, sure, he could check that out. Anything important you're going to keep under lock. The nucleus locks down the DNA. It also has this nucleolus, and this is made up of, so it's made of ribosomal RNA, and this is where that you make the ribosomal subunits. So it makes ribosomes. So of all the things here, we want to think about the nucleus. They always call it the control center. That's not really true. It's more like a vault. That's what it really is. It's a vault to hold the DNA. So I'm going to think about it like a vault. Very safe. Only certain things come in and out. The next piece over here, I know every time I move this it's loud and I hate that. So is the ribosome, and they're speaking about that because ribosomes are actually made in this nucleolus area. So there's ribosomes. We've talked about this before. They have two subunits. Large and small. Those are at least decent names. And they're made up of, ribosomal RNA. That's what they're made of. So they're going to be made up of that same, I don't know why I have to cough when I start doing this. Probably because I'm talking so much. But they're made up of the same 4 nucleotides that RNA has, but they're just called ribosomal, and it's had a different function. And so it's structural. And their job for ribosomes is where that proteins are made. That's their main function. Protein synthesis, we don't have to say all that. We can just be real clear with it. Then you are moving outside the nucleus. So we started in the middle and we're moving out, and we have that whole endomembrane system. The big components are going to be the endoplasmic reticulum. So this is that whole membranes that are in the cell, but outside, connected to the nucleus. So they're that membrane network outside of the nucleus. And there's 2 parts. There's the rough and the smooth. And so really structurally, they don't look very different, except the fact that the rough has ribosomes attached. But if you look at the membrane part of it, they're not that different. But they are different in their function. So we've talked about this before, and we could go see our notes. But to sum it up, the rough ER is going to have ribosomes, which equals proteins being made there. Also, membranes are made there. A lot of times the proteins are dropped into the membranes just like we had in the video right there. That's the big takeaway on the rough ER. The smooth ER, you know, I told you, you know, when they ask something about proteins, it's like, hello, go see my brother. That is not me. Yeah, we have the same last name, but we don't have the same first name. This is not me. I don't do all that stuff. The smooth ER is really different. Its job is to deal with detoxification. So it's going to detox, and it's going to make lipids, and then it has a really important job of storing calcium, which we turn around and let it go with a gradient, and it becomes part of signaling. So those are the big things. I feel like I'm forgetting something about rough ER. Carbohydrate chains, is that what, did I put that in there? No. Oh, is it on here? Did they say it? Oh, they do. Okay. So let's write myself a note and put glycoproteins are also there. So they modify them with carb chains. Okay. So now we're going to talk about Golgi. Golgi are still, that's like the apparatus there, is still part of that whole endomembrane system where that you've got all of this, but the structure of it are just, they call them like sacs. Cisternae is what their real name is, but they're, there we go. Stacks of flattened membranous sacs. Okay. So, stacks of membrane kind of sacs. That's what they really are. We drew this before. They have a sidedness to them. So the Golgi has, the cis is going to be the closest to the nucleus, and the trans will be the farthest from the nucleus. So that's just giving you name based on position. The job is, as membranes and proteins are sent to the Golgi, they're modified and they're packaged. So that's its big job. It's going to modify proteins and lipids, and then it's going to tag, and I'm going to put it like this, it's going to tag the proteins with carb chains. So what they're going to do then is become like the address label, because it's going to package all of this up in vesicles. So it's boxing it up and shipping it out to go where it's supposed to go. Examples of things that you get out of the Golgi. I hate the noise, I'm so sorry, it's not noisy here. One would be a lysosome. That's a specialized vesicle. So it's just going to be a membrane-bound vesicle with digestive enzymes inside. Often it has a low pH because these enzymes work best at a low pH. So it may have a microenvironment, is what that's called. So its job is to deal with waste. So it's going to digest the waste, and or it's going to digest anything that you, like even old organelles, materials brought into the cell. So it's all about waste processing. Then we have a vacuole, pretty small in animal cells, freaking-normous in plant cells. Like, no joke. If you've got a plant cell, here's the cell wall. The vacuole can take up all that room. There you go. I mean, it's the big thing in plants. It's all about water management. So this up here, lysosomes are about waste management. Vacuoles are about water management. So it's going to be the same kind of thing. It's just a membrane-bound vesicle with water. And its job is to mostly, if it's a water vacuole, it's going to be for water, but it can hold other things. Then it's not called a water vacuole, but it can, it's like storage. Maybe it's like a storage container. Most of the time they're for water. Then you have mitochondria. Number one structural component is double membraned because of the because of the endosymbiotic theory of where that it was engulfed and its own membrane is there and then the one that was around it. It has a really high amount of infolding of the membrane, which is going to increase the surface area. A lot of what happens on the membrane, so if you fold it, you have a lot more membrane. The entire job of cellular respiration is to break down sugar into energy. So that's cellular respiration, but that doesn't mean much to people. And do not ever say it's the powerhouse of the cell. Holy cow, people who read will flip out. Don't do that. It's going to break down glucose into energy that the cell can use. Pretty much the exact same thing, but backwards, is the chloroplast. Chloroplasts are so this is in animal and plant cells. Mitochondria are. Chloroplasts are in plant cells only. They're also double membraned because of that whole endosymbiotic theory where that it was engulfed, and have a lot of enfolding, and their job is photosynthesis. So they're going to take light energy and atmospheric carbon dioxide and make sugars. So the bond energy to bond that together comes from light. And then the last one of these is peroxisome. Goodness. I don't know why they didn't include that more up here, because it's also a membrane-bound vesicle. The difference it is, you know how we did the lab with the liver, and we had tons of catalase? The peroxisome is full of catalase. So its job is to take hydrogen peroxide with catalase, which is the enzyme that works on hydrogen peroxide. So this is the enzyme, this is the substrate, and it's going to make hydrogen peroxide not dangerous. It's going to change it to, change to water and oxygen. Okay, so there's going to be a video about this. The only thing that I would like to stress before I start with this piece is, and I'll put it over here, anything that is a double membrane, you want to have it in your head, you truly understand what they're saying. They are not saying it's a bilayer. All membranes are bilayers. Double membrane literally means two bilayers. So be careful that you have that in your head. So double membraned means you're going to have a bilayer, like right there, that's one, and another one also. So it's two bilayers. That's a big deal. If you don't have a bilayer, you don't have a barrier. So you could never have just a layer. If you took these three phospholipids and you put them in something by themselves, they would still orient tail to tail. So they're never going to exist into something like this, ever. There's no such thing as that. That will not exist. If you even had two, they would orient themselves tail to tail. So anytime you have phospholipids, they make a bilayer, and double membrane things have two of them. Okay, so we will, I don't know how long this was going to be. I can't make promises. But I'm going to try to make it within my budget. But I'm trying also to grab up all of the stuff that we have kind of hodgepodgey left over.
Eukaryote and prokaryote. Eukaryote cells are more complex in structure and function than prokaryote cells. In this tutorial, we'll tour the major features of a eukaryotic cell using both an animal cell and a plant cell as examples. Because of its large size, the nucleus is one of the most noticeable structures in the eukaryotic cell. The nucleus is the storage site for most of the cell's genetic information. The dark structure inside the nucleus is the nucleolus. This is where the components of the ribosomes are manufactured. The endoplasmic reticulum, or ER, is a membranous network of sac-like structures in the cytoplasm of the cell. The ER is divided into two distinct types based upon structure and function. The rough ER is studded with ribosomes, the location of protein synthesis in a cell. The presence of the ribosomes gives this ER its rough appearance. The proteins produced by the rough ER are often secreted by the cell and carried by vesicles to the Golgi apparatus for further modification. The smooth ER is a membrane-bound organelle involved in lipid synthesis, detoxification, and carbohydrate metabolism. It is given the name smooth endoplasmic reticulum because there are no ribosomes on its cytoplasmic surface. The Golgi apparatus consists of a series of flattened sacs that resemble pancakes. Its job is to package and distribute cellular products that are destined for a specific location within the cell or for secretion. You can think of the Golgi apparatus as the post office of the cell, where packages are dropped off and destinations are assigned. Lysosomes, which are produced by the Golgi apparatus, are vesicles that aid in the breakdown of materials within the cell. They may also digest worn or damaged organelles and recycle their components. Mitochondria are the location where the stored energy found in carbohydrates is used to produce cellular energy in the form of ATP. Mitochondria are found in both plant and animal cells. Unique to the plant cells are the chloroplasts, the location of photosynthesis. This is where incoming sunlight is used to convert carbon dioxide gas to carbohydrates and other compounds. Vacuoles are specialized membrane-bound structures found mostly in the cells of plants. Vacuoles usually store substances such as water, sugars, and salts, as well as pigments and toxic molecules. The pigments are responsible for the red, blue, and green colors of flowers and some leaves. The toxic chemicals keep the plant from being eaten by animals. Centrioles are organelles found mainly in animal cells. They are composed of microtubules and occur in pairs. Centrioles function in the reorganization of microtubules during the process of cell division. The cytoplasm contains a complex network of protein fibers called the cytoskeleton. These fibers include microtubules, microfilaments, and intermediate filaments. Overall, the cytoskeleton plays a similar role to the bones and muscles in your body, providing structural support, and along with motor proteins, allowing the cell and organelles to move. Let's do a quick recap. As we've seen, the internal structure of a eukaryotic cell is fairly complex and differs slightly between a plant cell and an animal cell. This diagram will help you remember some of the similarities and differences.
Okay, I feel like I need to wear some kind of armor because part of what we're going to do is water potential, and honestly, I think it's one of the most difficult things for people to wrap their head around. Luckily, we don't have to do a whole lot with it. In fact, I'm not even sure it's worth time, but we do give it a mention in case they throw a question in there. They're rare. So you have to think about the cost-benefit piece. The cost is unreal amounts of time for students to understand. The benefit, not so much. So I'm going to take this one. This is what we're doing now. This is called a U-tube. Not YouTube, but a U-tube. And they do have questions like this, which relate to water potential all the time. So we're going to do the ones that benefit us the most. And we will do some of these in class as well. So, I'm going to take my little guy in there. All of these problems have to do with a couple of things. 1, movement of water. 2, movement of solute, which half the time I call stuff. And 3, a semi-permeable membrane that lets some things through and not other things through. Kind of the definition of semi-permeable. So, I want to start down here. I don't want to do this. I want to build the point of this for you. So when they use a U-tube, it seriously is a glass tube that's been bent into a U, and it has a membrane right here. So this is the key part. Do I have a highlighter? Yes, I do. Right here. That it has a selectively permeable membrane. Then they're going to draw items that are suspended in this solution, which are called solute, stuff. And they're going to have different stuff. Some of it will go through the membrane because it's small enough. All of this is some type of barrier with holes punched in it, of a certain size. So big things can't go through, smaller things can go through. And then what happens is you leave this out, and it comes to, other word that's important, equilibrium, which means that everything is equally distributed on each side. Now that does not mean fair. What that means is whatever was able to move freely back and forth, you'll have the same amount of solute per liquid concentration as you do on the other side. Whatever was not freely allowed will also have come to equilibrium as far as the amount of water it takes to dissolve that. Probably made no sense. Easiest way to do it is to just do it. So I'm going to draw a U-tube. And I think I've talked to you about that pretty soon when you come to class, I'll hand you, it's usually a little foldable thing, kind of like this, where it's got one side and these two sides, and you fold it and put it in your notebook like that, like the bomb bath. And it's going to have some practice problems. This will become something that's on there all the time. So the first thing I'm going to put down here is my membrane that's selectively permeable membrane. And then you have some type of liquid, usually it's a buffer solution, and they can start with the levels of water at different places. We're going to start with them equal. Most of the time, they'll call the sides A and B. Then we're going to put stuff in there. So, solute. So I think what I'm going to do, and I hate doing it this way with the numbers, but I'm going to do it this way anyway, just because it's simpler. The red molecules I'm going to let be too big to go through. So I'm going to put twice as much of the molecule that's red in A as in B. And I'm visually representing that by 4 to 2. The green molecule, I'm showing this like you don't see it. The green molecules, I guess I'm thinking about Christmas, are going to be small enough to move through the membrane. So the membrane is permeable to the green. So I'm going to put, let's put like 4. I'm going to put twice as much in B as I did A for concentration. And I'm going to represent that by putting 8 green and 4 green. 8 in B, 4 in A. Okay, so right now we haven't talked about molarity, nothing. We're just saying, visually I can see there's twice as much of the red molecule in A as B, and there's twice as much of the green molecule in B as A. Okay? Then you let this go to equilibrium. So I'm going to let this go. So then I'm going to let this equilibrium. Now we're going to come back and talk about this in just a second, but I'm going to let it go to equilibrium. Sometimes in test questions they'll say something like, after 3 days. Well, everything that was going to happen definitely happened in that amount of time. Equilibrium just means whatever needs to move through the membrane, green molecules and water, because water is small, will have happened and will be done. So let's draw what this is going to look like when we're done. I'm not going to put water levels in yet. So let's start with the red. We will not have any movement of the red because I said that the red cannot move through. So if you had twice as much in A as you have in B, you'll still have that. So 1, 2, 3, 4. 1, 2. Okay? The green, we said those can move through. And I had 8 and 4, so twice as much in B as in A. So it would, at equilibrium, it would equalize across that, and I would have 6. 1, 2, 3, 4, 5, 6, and 6. 1, 2, 3, 4, 5, 6. I hope you can agree with me that that makes sense. Now let's think about the water levels. And I don't mind talking about this in very simple terms because it's very simple the way things work. Every single one of these molecules has to be surrounded by water. That's adhesion. Otherwise, it can't go into solution without its hydration layer, because then it's water with water, which is cohesion. That's how the molecules go into solution. That means that the water is busier, covering more molecules in A than it is B. Okay? That's one way to look at it. The other way to look at it is, just in terms of molecules, there's more molecules in A than there's B. A has more stuff in it. Water moves where the stuff is. So we're going to write that. Not very biology sounding, but it's exactly what happens. Water moves where the stuff is. So if you have more solute on one side of this tube than the other, you will also have more water. So I'm going to put what happens here. The red molecules, they did not move. So for the red, I'm just going to put that there was no movement. For the green moved from B to A. So the green moved in this direction. Because stuff moved to A, water moves to A also. So this would have an increase in the water level, and B would have a decrease in the water level. That's the kind of question they'll ask. After 3 days, what was the difference in height between the water levels? A is higher than B. Nobody's going to ask you how much higher. A is higher than B. Why? A has more stuff. That's an example problem. Let's look at this one. So I have to give the correct tonicity, that's an important word, for each side, and the correct solute concentration illustrated with dots. So I have to draw this out. So I'm going to draw it. When they don't tell you what is able to move, then you've got to start looking right through here. They're telling me in that terrible little copy that water can move. They are not saying, and nowhere did they say, that these dots, these molecules, this solute can move. If it could, you would have movement of the dots from this B side. So this is A, this is B. From B to A until it would be equal in the number, and water would move with the stuff, and you'd have a higher level in A. But that's not what they're saying. Nowhere did they say these molecules can move. So these dots cannot move. Only water. Water moves where the stuff is. So the concentration of solute on this left side in A is much lower. You can see it visually. There's less stuff in the water than over here in B. Water will move until the concentration, the ratio of water to molecule is the same on both sides. So what will happen, draw my membrane, I'm not worrying with dots. They only asked me for the correct water level. So the water will move in this direction, and I will have more water on the B side. So initially, let's look at tonicity. Tonicity has to do with words. It has to do with hypertonic, more stuff. So more solute, more stuff. Hypotonic, like hyper, you can think about that. You know what it is to be hyper. Like you are way up. You're extra. You're like crazy. High. Hypo, think about a hypodermic needle that they put under your skin. So this is low. This would be less solute, less stuff. Well, whenever you use the words more and less compared to what? Both of these are relative. That's a huge word. You can never say something is hypotonic. It has to be hypotonic compared to something else, which means it has more stuff than something else. There's another word that's called iso, which means same tonic, and that would mean the same solute. And they're talking naturally about concentration. So let's go up here and let's put words up here. You probably can't even read those. There's less stuff in the same amount of water in A compared to B. So I would write A is hypotonic to B. A is less stuff compared to B. I could say B is hypertonic, more stuff compared to A. This is going to be in this figure here. We'll call it 1. After 3 days at equilibrium, whatever you want to call it, the water will have moved until there is the same amount of stuff per amount of water on both sides. So this is going to be at equilibrium, which is 2. You are isotonic to each other. A and B are isotonic. They have the same concentration of stuff to water because the water moved. That's the kind of questions that you have to be able to do. And we will do many, many more. And I have a video after this. And I don't know. I like Paul Anderson. And if you know Miss Lupe, if you ever knew who she is, if you had her at Robinson, I don't know if y'all are like old enough to have had her at Adams. But anyway, she's fabulous. She's in Saudi right now. Her and Mr. Lupe, John, who taught, what did he teach? PE at Washington. Anyway, I'm telling you this because she sends me pictures with her and Paul Anderson, like all buddy-buddy. So her people in Saudi, where she teaches, they fly him to Saudi to do professional development. Whoopie-doo. You are fancy smancy, Kathy Lupe. But I'm going to put a Bozeman video here because he does walk you through this. We will do problems. The more you do, the better you get.
To demonstrate how tonicity affects a cell, let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cells, the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking, of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment. In other words, water moves toward areas of high salt or sugar concentrations. This simple process is used to drive the operation of our kidneys and explains some of the physiological consequences of diseases such as diabetes.
When we were kids growing up in West Texas, our winters would be cold but rarely experienced snow. But we did have ice, which resulted in the roads being salted. As the salt mixes in and dissolves into water on the road, this can lead to a lower freezing point, which can help prevent the roads from icing over. And while this is great for making the roads more safe, it wasn't so great for the plants that lived right along the roadside. It often caused them to die. Now, winter can be hard for many plant species, but I'm talking about this salt affecting even some hardy plant life. This issue with salt in plants isn't limited to winter. During hurricanes near the coast, salty ocean water can be dumped in large quantities into the soil. This can eventually kill plants, including trees that had originally survived the hurricane. Why? Do plants just dislike salt that much? Well, it's actually related to a term called osmosis. When you are talking about osmosis, you are talking about the movement of water through a semi-permeable membrane, like a cell membrane. Water molecules are so small that they can travel through the cell membrane unassisted, or they can travel in larger quantities through protein channels like aquaporins. The movement of water molecules traveling across the cell membrane is passive transport, which means it does not require energy. In osmosis, water molecules travel from areas of a high concentration of water molecules to a low concentration of water molecules. But there's another way to think about water movement in osmosis. A low water concentration likely means there is a greater solute concentration. Solutes are substances like salt or sugar that can be dissolved within a solvent, like water. Water has the tendency to move to areas where there is a higher solute concentration, which would mean less water concentration. So if you want to easily figure out where the water will travel in osmosis, look to the side where there is a greater solute concentration. Unless we bring in another variable, like pressure, water will generally have a net movement to the area of higher solute concentration. So let's bring out a U-tube. U-tube, that's funny. There's a semi-permeable membrane in the middle of it. Let's assume that it is similar to a cell membrane and that water molecules can squeeze through it. The molecules are quite small, but salt can't. Right now, there's just water in this U-tube. The water levels on side A and side B are equal. That doesn't mean that the water molecules aren't moving. Water molecules like to move, but the net movement across the two sides is 0. That means the overall change in direction of movement is 0. Now, let's imagine on side B, you dump a huge amount of salt there. So which direction will the water initially move towards? A or B? Think about what we mentioned with osmosis. The answer is B. Side B has a higher solute concentration than side A. Water moves to areas of higher solute concentration, which is also the area of lower water concentration. You will also see the water level on side B rise as the water moves to that area. You can almost think of the water as trying to equalize the concentrations, diluting side B. Once equilibrium is reached, the net movement of water across the two sides will be 0, but remember that water still likes to move and movement still occurs. Now here's some vocabulary to add in here. We call side B hypertonic. That means higher solute concentration. But we can't just say something is hypertonic without comparing it to something else. We say side B is hypertonic to side A because it has a higher solute concentration than side A. In osmosis, water moves to the hypertonic side. We say side A is hypotonic. Hypo rhymes with low, which helps me remember that it's the low solute concentration when compared to side B.
Hi, it's Mr. Anderson, and today I'm going to show you a quick little osmosis demo that I set up in my classroom. But before I do that, I should probably talk a little bit about what osmosis is. And so osmosis is simply diffusion, or the movement of water from an area of high water concentration to low water concentration across a semi-permeable membrane. So what does that mean? Well, if you look right here in this U-tube, that's what this is called, this would be a semi-permeable membrane. And what that means is it only allows a few things to move through. And so you can see right here that the water molecules are small enough that they can pass through, but the solute molecules aren't. Solute molecules could be sugar, could be ions, but essentially you can't make it through. They're too big. And so in relation to this side, the left side here we would call the hypertonic side. What hypertonic means is greater levels of solutes. And that means over on here, this in relation is going to be called the hypotonic solution. In other words, there's greater solute concentration on this side than there is over here. And so these are in relation to each other. And so what does that really mean? Well, if we were to zoom in at the molecular level, all of the water molecules are going to actually stick to this solute. So the water molecules are going to grab onto that. And since the water molecules are grabbing onto the solutes, there's not going to be a lot of water on this side. And so we're going to say that the hypertonic side is actually going to be an area where there's low water concentration. So over here, there's less solute, so there's less water molecules grabbing onto the solutes, and so the water is going to flow in that direction. And so however you want to think about it, is the water flowing from an area of high water concentration to low, or is the water flowing towards a hypertonic area? Either of those, it doesn't matter as long as you know which way the water is going to flow. And so what's going to happen to the water? Well, the water is eventually going to flow over to the side until the concentration of water on either side of that semi-permeable membrane is exactly the same. At this point, we would say on either side, it is now isotonic, if I can spell that right. Isotonic means that water is still flowing back and forth, but it's flowing back at an equal rate. Now, if you were to think about this, if you were to watch this, it would seem somewhat magical as the water is just moving up on this side, just kind of against gravity and dropping on this side because you can't see the solutes. And so that's pretty cool. A
semi-permeable membrane, also called a selectively permeable membrane, is a membrane that allows certain molecules or ions to pass through it while blocking others. One example of a semi-permeable membrane is a phospholipid bilayer, a group of phospholipids consisting of a phosphate head and two fatty acid tails, arranged into a double layer, with the hydrophilic phosphate heads exposed to the water content outside and within the cell, and the hydrophobic tails hidden on the inside. In general, small, non-charged molecules such as oxygen or carbon dioxide can freely cross the membrane without an input of energy. They're able to slip between the heads of the phospholipids and pass through the hydrophobic tails through a process known as diffusion. These molecules are able to move down their concentration gradient as they move from an area of high concentration to an area of lower concentration. In contrast,
Okay, so there's no glue in for this one. This is just a concept that comes up a lot, and we want to be sure we talk about it. It's not anywhere in our glue-ins or notes or whatever, but it's a process called cell fractionation. This just means you broke the cell. That's what that means. So this is a process where that you can separate out, so let's put that, so a process to separate cell organelles, and it's going to be by using a centrifuge. Centrifuge works by spinning things, and what happens is when you put it in the centrifuge, whatever is heaviest and biggest moves to the bottom. So you separate by size and mass. So that's the whole point of this. And you do a series of these. I used to do them all the time in the lab. Nobody who studies something, like I studied membranes. I don't want all the other stuff. I just want to study the membranes of the cell. Everything else, I don't want it. Well, cells don't really just offer up their parts. You have to separate them. And luckily, you can do this by using the centrifuge, and what you'll have is a series of spins. So there's going to be some words I use, and let me explain. So every time you run the centrifuge, you're spinning. That's what it's called. You'll do a spin. A lot of these centrifuges are about the size of a refrigerator or bigger, laying flat. They're heavily armored because when they spin, they can spin so fast that if that whole centrifuge case that you put your tubes in got out, it could take its way through walls. It's got that much force associated with it because of the spin. Plus they get really hot, the friction from the spin, so they have to be super cooled, and that's why that big case is there, so they keep them cool. They're expensive too, like you have to sign up to use them. So what you're going to do is you're going to do a series of spins. Every time you spin, you have something that you have caused to move to the end of the tube, the bottom of the tube. That's called throwing down. So when you have a spin, you throw down of whatever the liquid was that was in there, whatever has the most mass and the largest size. So that's what these words are. So if I say spin and throw down, that's what I'm saying. So the very first thing you're going to do, so we're going to put this in here, you're going to separate by 1 mass and 2 by size. So whatever you're separating, these organelles, these pieces, that's what's going to be done when you use the centrifuge. So the very first thing you have to do, it's kind of creepy, you put cells in a blender with a buffer solution. The solution is going to keep the pH correct so that you don't have the unfolding of all the proteins, and you're not going to have things change in their shape. So you're going to have your cells plus a buffer solution, and you're going to put that in a blender, and you're going to blend it up. No joke, like a Ninja blender or a Vitamix. A regular blender from your kitchen is what everybody uses. Once you have them broken up, you're going to start your series of spins. So you're going to have, this is your spin, number, the first spin is going to be with everything in it. So it's all of the cell parts. So spin 1, you're going to have, this is what we've got in the actual tube. So we're going to put in here, we're going to have broken up cells, because that's why we put them in the blender, in the buffer solution. You're going to take the tube and you're going to spin. There's your job. Go spin. You don't spin it very fast. You don't spin it very long. You're just wanting to throw down the big chunks of stuff. So this spin is slow and short. It's got a short period of time. When you're done, what you're going to end up with is, in your tube, you'll have the solution and whatever was big and whatever biggest or the most mass will be thrown down to the bottom, and this is called a pellet. So the pellet contains the largest of whatever's in the solution. So you throw down this. The liquid that's left is called the super, which means above, natant. Supernatant. Forgot where it was. So you're going to see a pattern in how this works. I did a spin. It had everything. I did the spin slow, short. What I threw down in the pellet are the big things. Think about what that would be in the cell. This pellet is going to, the pellet has the nuclei from all of those cells. If that's what you're studying, you scoop the pellet out and you throw everything else away because you have what you wanted. The supernatant has everything else. Not the nucleus, everything else. Well, but I don't want the nucleus. Okay, throw the pellet away and let's spin this solution. Okay, so if you don't want the nuclei, you're going to do spin 2. In spin 2, you take the supernatant from spin 1. It's going to have everything but the nuclei. And you spin again. So whatever in there that's left, that's large or heavy, we'll spin it down, but we can't spin it down slow in a short period of time. The smaller something is, the longer and the harder, faster, you have to spin it. So we're going to go faster, right, and for a longer period of time. So this time you're not going to be able to spin short and slow. You're going to turn the centrifuge up, and you're going to let the tubes spin longer. When you're done, you get the exact same, the conformation, the way it looks. You'll have a pellet. There's always a pellet and a supernatant. This pellet now is going to contain whatever was next biggest to nuclei. So the pellet has mitochondria and chloroplasts. So if we're ranking organelles in size, spin 1 gets the big guys, nuclei. Spin 2 gets the next big guys, mitochondria and chloroplasts. And whatever is left in the supernatant, you know what I'm going to write here. Supernatant has everything else. not the nuclei, not the mitochondria and the chloroplasts, but everything else. Well, but I don't want the mitochondria and the chloroplasts. Okay, get rid of your pellet and let's spin again. So if that's not what you want, you didn't want nuclei, you didn't want mitochondria and chloroplasts, this is me. Okay, I didn't want any of that. I wanted membranes. So I would take the supernatant, go for spin 3. Here it is. It's everything in here now except the nuclei. Those came out in the first, they were thrown down in the first spin. And it doesn't have the mitochondria or the chloroplasts. Those were thrown down in the second spin. And I'm going to spin again. You'll get the pattern. This is what's left in here is smaller stuff. So I'm going to have to go even faster as far as the number of spins per minute and for an even longer period of time. As you always do, you throw down a pellet and you have solution that's left, supernatant. This pellet has membranes. This is where I stopped. I grabbed the pellet, throw everything away. That's what I wanted. If you did not want the membranes, or the mitochondria, or the chloroplasts, or the nuclei, because they're long gone, they were these pellets, and we threw those away a long time ago, then you're going to have to spin again for what's left. And there's only one thing left. So this supernatant has everything else that's left. We'll spin it again. Because if you're someone who studies ribosomes, you're going to spin one, and what you've done is you've culled out, which means you've pulled out and thrown away the nucleus. Spin again, you cull out the mitochondria and the chloroplasts, throw them away. Spin again, cull out the membranes, spin again. So this is our fourth spin. And this supernatant solution has everything. It sounds funny because we've taken most of it out, except the nuclei, the mitochondria, the chloroplasts, and the membranes. They're all gone. Those were in the pellets. Now what we'll do is we'll spin, and you spin for freaking ever. Like this when you sign up for the centrifuge, people hate anybody who's going to do for ribosomes because it goes forever. So it's going to be a really long, now let's go with really fast. A really fast, like speed. And it's going to go for a really long time. So you're going to spin ridiculously fast, and you're going to let it go forever. Like it feels like forever. When you're done, you always get the same thing. You're going to have a pellet, and the pellet is going to contain what was solid and left in here. So the pellet has the ribosomes. So they're the smallest. And your supernatant really is only going to have just the cytoplasmic type. So supernatant. It's going to have more of the cytoplasmic liquids, like just the stuff that's still not throwdownable. So sometimes you keep the pellet, sometimes you take the supernatant and respin. But let's summarize what we found, and I'm going to put it right here, just so we can make it. We're going to put, here's my spin. I had 4, right? 1, 2, 3, 4 spins. Spin 1, I got, if I've used it and it stopped, this is where I would get the nuclei. That's what you get after spin 1. And we'll think about the rest of this in just a second because I want to talk about size and speed. Spin 2, what you could get would be your mitochondria and your chloroplasts from spin 2. Spin 3 are membranes. Spin 4 are ribosomes. So what you're going to notice about what you were able to spin out is by size, this has a larger, a higher size down to the lower, smaller size. Mass, the nuclei have more mass, so their mass would be higher, and as you go down this line, ribosomes would have the lowest mass. This is inversely proportional to what you do with the machine. So for the machine, when you centrifuge, big things, high, have low speed. If you want the smaller things, you got to crank the machine up. Your speed has to be high. And your time also is inversely proportional. When you're doing nuclei, they're huge. Their mass is huge, their size is huge. You don't have to use much speed or time, so your time amount's low. If you want something tiny, like ribosomes, you're going to spin hard, which would be high speeds, for a long time. Long period of time. Okay? So, questions that they ask you about what would you do in order to get certain components out. So always think about size is the easiest way to remember what's thrown down. Okay? Alright, so I have a little video for you to watch with that.
Since we know cells are made up of a lot of different parts, it's useful to understand what each part does as well as their individual structures. In order to do this, you have to be able to study each part separately. A process called cell fractionation was developed to help with this. It takes cells apart and separates the major organelles and other cellular structures from one another. Tissue cells are first homogenized, or broken apart. Their plasma membranes are broken up so that their internal contents spill out and mix together in what we call a homogenate. The homogenate is then spun at a high rate of speed in a process called centrifugation, and that speed can be varied, so we call it differential centrifugation. Depending on what speed you use, different parts of the cell will accumulate in the bottom of the test tube in a pellet. The reason for this is because different parts of the cell are bigger or smaller, and the bigger they are, the quicker they'll sink to the bottom, so you don't have to spin it as fast. Whereas other pieces are very small and you have to spin it at a very high rate of speed to force them to the bottom of the tube. For example, if we first spin the tube at something like 1,000 Gs, or 1,000 times the force of gravity, you'll get a pellet rich in nuclei and cell debris. But there'll still be a liquid portion above that pellet called the supernatant. And within that supernatant, there'll be other parts of the cell that are smaller. If we pour that supernatant off and spin it at a higher rate of speed at something like 20,000 Gs, you'll get a pellet rich in mitochondria. If you repeat that process and spin that supernatant down at something like 80,000 Gs, the pellet will be rich in microsomes, or pieces of plasma membrane and internal membranes. And if you spin that supernatant down at 150,000 Gs, you'll get a pellet rich in ribosomes. This technique allows for the isolation and subsequent research of subcellular components so that you can determine more about their individual roles in the cell.